In the previous videos, I described the cause and mechanism of PKU. Much less is known about the link between PKU and mental impairment. In this video, I'm going to explore the various possible causes of mental retardation in untreated PKU patients. As you recall, inactivation of the enzyme pH in PKU patients prevent the conversion of phenylalanine into tyrosine in the liver. Consequently, phenylalanine accumulates in the blood and saturates the large neutral amino acid transporter at the blood-brain barrier. The excess of phenylalanine and the deficiency in other large neutral amino acid in the brain leads to oxidative stress, a decrease in neurotransmitter synthesis and lipid metabolism, bioenergetic deficiencies, and an excess of intracellular calcium. Let's first discuss the impact of PKU on oxidative stress in the brain. Oxidative stress is caused by an elevated level of oxidants, including free radicals, formed when some molecules react with oxygen. Free radicals contain chemical groups with atoms characterized by an odd number of electrons. These free electrons are available for pairing, making free radicals very reactive molecules. The oxidative damage caused in cells by free radicals can be prevented by molecules called antioxidants. By donating electrons to free radicals, the antioxidants neutralize the oxidative effects of free radicals. The most important group of free radicals in biological systems are called reactive oxygen species, or ROS. ROS are the natural byproducts of cellular metabolism. At low concentration, ROS are essential for several physiological processes. Under normal conditions, their concentration is maintained low by the presence of antioxidant. The redox state of the cell is maintained at equilibrium. If the production of ROS overwhelms the antioxidant capacity of cells, the imbalanced redox state leads to oxidative stress. The highly reactive ROS start a chain reaction like dominoes falling. Essential cell components such as DNA, proteins, and cell membranes are damaged. The functions of cells are impaired, and some cells might die as a result of oxidative stress. Oxidative stress has a dual cause. The PKU diet is partially responsible for oxidative stress. Some food rich in antioxidants cannot be ingested by PKU patients, and nutrient deficiencies affect the synthesis of endogenous antioxidants. The disease itself causes oxidative stress by tipping the balance between ROS and antioxidants toward ROS. The chronic exposure of high concentration of phenylalanine and phenylalanine derivatives triggers the production of ROS. Furthermore, at high concentration, phenylalanine inhibits the production of antioxidants. What are the consequences of oxidative stress in the brain? Signal transduction and the function of a type of brain cells called astrocytes are impaired. Brain inflammation is observed. An imbalance in superoxide level is also observed. As a result of this oxidative stress, abnormal neuronal cell death and aging are observed in PKU patients. PKU also inhibits the synthesis of neurotransmitters. The effect on the synthesis of neurotransmitters is due to the accumulation of phenylalanine and to a decreased level of tyrosine in the brain of PKU patients. Tyrosine is the precursor of a group of neurotransmitters called catecholamines. We know that tyrosine needs to be transported into the brain across the blood-brain barrier. All the large neutral amino acids, including phenylalanine, tyrosine, and tryptophan, use the same transporter. Therefore, an excess of phenylalanine outcompetes tyrosine and tryptophan for the transport into the brain, leaving the brain with a reduced tyrosine and tryptophan concentrations. Tryptophan is a precursor of the neurotransmitter serotonin. As you recall from a previous video, after being transported in the brain, tyrosine is first converted to dopa, which is in turn converted to dopamine, norepinephrine, and epinephrine. Therefore, a reduction of the concentration of tyrosine negatively affects the synthesis of catecholamines. Not only is the brain supplied with less neurotransmitters precursors, but phenylalanine is a natural inhibitor of the enzymes responsible for the synthesis of L-dopa and dopamine, 5-hydroxytryptophan, and serotonin. Therefore, the excess of phenylalanine in the brain of PKU patients further reduces the production of catecholamines and serotonin. What are the consequences of a deficit in dopamine and serotonin? The lack of dopamine is responsible for Parkinson-like symptoms and an inability to feel pleasure called anhedonia. Lack of serotonin is responsible for OCD-like symptoms and uncontrolled impulsivity. In combination, a deficit of these two types of neurotransmitters is linked to depression. The lipid metabolism deficiency in PKU patients seems to be linked mostly to the dietary restrictions. The diet is very restrictive, especially in the early years, and PKU patients consume very little cholesterol and DHA fatty acids, a long-chain form of omega-3 fatty acids. The result is a low level of cholesterol and DHA in the body of PKU patients. DHA is the most abundant polyunsaturated omega-3 fatty acid in the brain and the retina. By weight, it represents 50% of fatty acid found in the plasma membrane of neurons. DHA plays important roles in neurons, including the transport of metabolites such as choline or glycine, the activity of potassium channels, the response of rhodopsin contained in synaptic vesicles. It is not surprising that a deficiency of DHA is associated with cognitive declines. Low level of DHA lowers neuronal activity and increases cell death. Low level of DHA is also observed in the brain tissue of severely depressed patients. DHA is also an important component of myelin sheath that coat the axon of some neurons. Myelin forms an electrically insulating layer that facilitates and speeds up the conduction of action potential along axons. Defect in myelin sheath caused by DHA deficiencies can lead to cognitive disruption, various neuropathies, difficulty in controlling movement and imbalance. It could explain, in part, the Parkinson-like symptoms in some PKU patients. The brain consumes a lot of energy in the form of ATP and rely most exclusively on imported glucose as a fuel source. High levels of phenylalanine decrease the efficiency of several ATP synthesis mechanisms in brain tissue. As you recall from a previous video, the creatine-phosphocreatine system serves as an energy reservoir for the rapid buffering and regeneration of ATP. Indeed, the interconversion of creatine and phosphocreatine is coupled with the interconversion of ATP and ADP. This system is essential in tissues that consume a lot of ATP, including the brain and muscles. The rapid synthesis of phosphocreatine needed for the fast regeneration of ATP from ADP is catalyzed by an enzyme called creatine kinase. This enzyme is inhibited by phenylalanine. Therefore, high levels of phenylalanine in PKU patients disrupt the creatine-phosphocreatine system and affect ATP synthesis efficiency in the brain. As you recall, the last step of glycolysis is the conversion of phosphoenol pyruvate into pyruvate and is coupled with ATP synthesis. The enzyme that catalyzes this reaction is called the pyruvate kinase and is inhibited by phenylalanine. Therefore, the yield of ATP synthesis by glycolysis is reduced in PKU patients. The brain is not so much affected because most ATP in the brain is synthesized by oxidative phosphorylation in mitochondria and by the creatine phosphocreatine system. However, it is important in red blood cells that rely exclusively on glycolysis for the ATP synthesis. The red blood cells of PKU patients have 5 to 25% of normal pyruvate kinase activity. The low rate of ATP synthesis prevents red blood cells to maintain their structural integrity. These cells lose their shape and are actively phagocytosed. That would explain the anemia observed in PKU patients. Under fasting conditions, the blood sugar level is low and the citric acid cycle slows down. Liver cells adapt their metabolism by producing ketone bodies that become a main source of fuel molecule for the brain. The synthesis of ketone bodies is decreased by high level of phenylalanine. Therefore, in PKU patients, the availability of this alternative source of fuel molecules is impacted and contributes to a decrease of ATP synthesis in the brain of patients. Finally, the level of free calcium in neurons is affected in PKU patients. Calcium plays an important role in different processes: neuronal excitability and plasticity, axonal transport. Low and high levels of calcium affect neurons' activity. Calcium is transported into neurons by membrane-bound transporters. Let's first describe the role of calcium in the formation of an action potential. Upon neuron stimulation, the cytoplasmic concentration of free calcium spikes. It is due to the activation of the ligand-gated calcium channel, moving calcium from outside to inside neurons. It's also due to the activation of the sarcoplasmic reticulum calcium ATPase, or SERCA, that releases in the cytoplasm calcium that was trapped in the sarcoplasmic reticulum. The spike of cytoplasmic calcium triggers neuronal firing. However, once the neuron has fired an action potential, the cytoplasmic concentration of calcium has to go back to a normal level. If the calcium level remains elevated, the function of the neuron is affected. The decrease of the concentration of free calcium is achieved by the activation of the plasma membrane calcium ATPase, or PMCA, exporting calcium outside of neurons, and the activation of SERCA that this time will move calcium from the cytoplasm to the sarcoplasmic reticulum, where this calcium is stored until the next neuron firing. Studies of neurons in culture allowed scientists to propose a model explaining how an elevated level of phenylalanine can affect neuron function. First, a high concentration of phenylalanine transiently activates PMCA. The consequence is an increase in calcium efflux and thus a decrease of the cytoplasmic concentration of free calcium. The triggering of action potentials is affected because the spike of cytoplasmic calcium is harder to achieve. Second, a longer exposure to a high level of phenylalanine has a chronic effect on neurons. PMCA now becomes inactivated. Calcium efflux is impaired. Neurons have a sustained high concentration of cytoplasmic free calcium. The triggering of action potential is affected. Overall, by affecting the modulation of the concentration of free calcium in the cytoplasm of neurons, an elevated level of phenylalanine causes neuronal injury. In conclusion, the elevated level of phenylalanine in the brain of PKU patients affect brain function in a variety of ways that could explain why, if remained untreated, PKU patients exhibit cognitive abnormalities. Many studies remain to be done to further our understanding of the correlation between PKU and mental retardation.